Okay, so this is a shortened title of the talk, Quantum Effects in Biology, question mark. I will introduce some of these experiments that Wayne alluded to, but really what I want to do is set anybody up with a more general background just to understand where some of the recent breakthroughs came from, why there are questions surrounding all of this and what those questions are. So that's really what I want to get to, rather than getting into a lot of detail with some of this technical stuff. To just calibrate everybody, why we care about coherence type phenomena is, order is everywhere, and synchronization matters for all of this in many different ways. So all the many thousands of cells in the heart fire together, and that's what makes the heart beat. This collective phenomena obviously is important for us, and it's this synchronization, which is a simple mechanism, but non-trivial phenomenon, and it's everywhere on the macro scale, on the micro scale, and it's on the micro scale where it's a bit more difficult to pinpoint, to grasp, and certainly to measure. So on the left, there's an example of synchronization. It's a sound wave from an orchestra playing. So this is what we would call coherence. And we can tell by listening what is coherence and what is white noise, for instance, very easily. On the scale of chemistry, what we often think about is that reactions and other processes happen stochastically. So more in the white noise regime, which is what I've represented there on the right-hand side. On the other hand, we understand that at the smallest length scales, coherence makes chemistry as a science. This is how you bring atoms together to make stable molecules, for instance. It's the superpositions of orbitals. So where else can it be exploited? And this is the question. The issue is, as we go beyond a molecule to interactions between molecules, is there any kind of synchronization or choreography there that involves quantum mechanical effects? Why we would care about this is because, as you know from engineering on the human length scale, if we want to optimize process or control processes, you do need effectively synchronization. You need all the transistors in a computer to be doing whatever they're doing at the right time, not randomly. So if we want to control or direct processes or optimize them, then some kind of synchronization or order is important. When we look at the molecular scale, obviously we can't see things of this nature by our eyes. Experiments are important. And one of the issues is that averaging often hides what we'd like to see. So if we do an experiment of the kind I'll show you in a moment on proteins from photosynthesis, have to remember that we're probing probably something like 10 to the 18 proteins in a small volume of solution in that experiment, and taking an average over those proteins. Even if we studied one of those proteins individually, which is possible in certain experiments, we're still taking that same average by the ergodic principle, because we have to probe it 10 to the 15 times to take an average. And so we're looking then at the average as the protein samples different conformations, energies, etc. in time, and it amounts to the same thing. So yeah, why the zebra? Obviously, if we can image something at the relevant scale, we can see that the zebra has stripes. But if we do some kind of experiment that takes an average, then we would think the zebra was gray. So this is where it can matter. That's a kind of obvious example. The picture of the water, snapshot of the water simulation at the bottom there says, well, it looks like a lot of things in chemistry might be random. Water, for instance, is a liquid. Is that not random? No, there's correlations on a number of length scales, even in a liquid, that we can measure, and that's been known for a long time. So there are correlations even where intuitively we may not expect. And a final example which illustrates both surprising correlations and also the ability to measure them if we do the right experiment is it's been known for hundreds of years that if we look at celestial bodies like the rings and Saturn from the Earth, they're twice as bright to the observer if we look at if we make the observation when the sun is directly behind the Earth and we're looking out away from that towards the planet. Why is that? Well, it's a phenomenon called coherent backscattering. And while just with this picture I'll show you, in your head, you can't work out why this is so, but I can promise you, you can easily demonstrate this, is that when the sun is at any angle in the sky, its light hits the rings of Saturn, and that light is reflected back to us. It's reflected back after trillions of reflections off the ice particles on the rings. So it's scattered back to us. When we do the measurement, if the sun is behind us, some of those back reflections will follow exactly the same path as when the light went through those ice crystals. They'll come back on exactly the same path. Those waves will lie on top of each other, and you'll get constructive interference when you measure the intensity. And this makes a difference, actually, to a macroscopic observation, that simple change in how you do the experiment. And this kind of experiment can be done in the laboratory as well. So that's just a little bit of background just to say, what is coherence? Why are there challenges when we start to look at this at the length scale of molecules, not even talking yet about the complexities of quantum correlations? And that the fact that doing the right experiment, a subtle change in how you do the experiment, can make a big difference. And that's typically about averaging, the issue of averaging. So really what I want to get to here is there's going to be a theme of coherence and entropy that I'll mention. I will give some background on the photosynthetic light harvesting and those experiments to give you a sense for what was done and what was the breakthrough in doing the experiment. I'll give you an intuition for the experimental approach here after that, an example where that actually works well in a simple case. And then we'll get to the question here. At the end of the day, we're trying to beat entropy. But to start with, I'm going to show you something completely from left field that I threw in here because it's a dramatic example, and it illustrates a few things just to get us on board with the kind of coherent phenomena that people have been looking for specifically in the light harvesting proteins and molecular systems too. And that coherence is, let me put it this way. If we have many molecules in solution or in a plastic film in this case, and you can see on the left in the red, the absorption spectrum of those molecules, what's happening there is that each molecule is interacting with the radiation field weakly, and there's some chance each one will absorb light. That probability does not depend at all on any of the other molecules in solution. So we individually add up each of their absorptions, and you see, well, I can't point to it, I guess, or maybe I can, I'm not sure. No. But on the left, where that blue little bar thing is, on that shoulder, that's the first absorption band of this particular molecule. Now, what we can do to illustrate the coherence in a couple of ways here, if we put that film essentially between two mirrors at a distance that's proportional to the frequency or the wavelength of that transition, we make a cavity. And what we do something strange actually in that cavity is we change the allowed or the amplitude distribution of the allowed modes of the electromagnetic field is what really happens. What it means is when each molecule couples weakly to the radiation field, it couples to a single mode, like a single harmonic oscillator in the ideal case. And it turns out then that all the molecules in solution couple to that same oscillator. So when one of them absorbs light, it's a little bit like the light is a steel bar attached to all the molecules, and we're driving them all together, etc., driving their transition dipole together synchronously. And when they absorb light, they do it collectively. In fact, remarkably here, about 100,000 molecules absorb light together in complete synchrony. And what does that mean? You know, what's how do we see that? On the right-hand side, it looks like a completely different absorption spectrum. It's the same system, but in that cavity, and you see that sharp, this is at different angles, but you see that sharp absorption band on the right-hand side. It's hidden by pictures of you on my screen here. That is the shift of thousands of wave numbers that's indicating that collective absorption of the molecules. So they're synchronized quantum mechanically, and that's in fact called a polariton state because they're all synchronized by the radiation field. I'm going to show you examples where they synchronized because they're attached to each other through an electronic coupling. And that's what we'll be looking for in many of these experiments. So this is the level structure picture, the ideal picture of what I showed you here. I think we could just look on the right-hand side. That band that's split off there is called the lower polariton in this state, in this picture, and it splits off down. It's completely coherent, very low entropy. It takes work to synchronize molecules. This is a very low entropy state. It's not where that system wants to be. And that's the challenge here. Whenever we make these coherent states, the entropy is low. And as you know, as time goes forward, entropy increases. And that's what we've got to beat. So that's the summary here. And in these cavity experiments, which I won't go into, we can do direct comparison because we have both the low entropy polariton band I showed you and a bunch of dark states, 10 to the 5 minus 2 dark states here that have the entropy of molecules in solution, essentially. So we can compare quantum states, and that's something I think that's an opportunity for the future. Here, I'm going to show you an experiment that would just be, well, how can we do an experiment that might identify that synchronization after light absorption or after any process? In fact, we now know that's kinds of synchronization like that. We have a recent paper coming out that shows it can evolve in time after we start the experiment. So photosynthesis, we all know the basics of this. Often we'll write it as one equation in a high school. It's really, to be fair, two equations. There's two enzymes here at the heart of this. One splits water. One reduces carbon dioxide. Each one of these enzymes is powered by light absorption. They take 4 photons each, but they're coupled, ratcheted together by the flow of electrons. So these are solar cells, in a sense, these enzymes. But what's interesting about them from a physical chemistry point of view, if you like, is if we take a leaf, separate the photosynthetic proteins, as in a sucrose gradient shown here, disperse them by molecular weight. If you look at the lower part of that tube, a little green band there highlighted with the lower arrow, that's the solar cell. So it's actually photosystem 2, the heart of that water splitting enzyme. There's not a lot of green there compared with the green in that upper band. So it seems like, unlike what we might imagine a solar cell to look like, there's other proteins that the plants think are more important in terms of the number of them for the function. What are those proteins? The proteins in plants anyway that look like this, it's 45 chlorophylls jammed into this protein at incredible concentration. In fact, it's so concentrated, that chlorophyll, that if we made a solution at that concentration, that solution would not fluoresce at all. In other words, it would be a great light absorber, but that energy would be lost as heat in a picosecond. We couldn't do anything with it. Plants somehow have managed to jam that many chromophores into a protein. A chromophore is a light absorbing molecule, and be able to retain a long excited state lifetime of a few nanoseconds so that they can do something with it. So what do? Clearly it's implicated in the absorbing light. And this picture shows a map of what happens is that any chlorophyll and these proteins that flank the enzyme that does the chemistry can absorb the light. And then what happens is that energy from the light needs to jump from chlorophyll to chlorophyll. This will happen 1,000 or 2,000 times until it reaches the enzyme, the reaction center, and sensitizes that. And then we get the flow of electrons. And what has amazed biophysicists here is that this, after excitation of light, 90% of the time, that excitation does in fact initiate electron flow at the reaction center. So despite this apparent random walk of excitation, jumping from chlorophyll to chlorophyll, it gets there. And so people have wondered, is there something more behind that? And why do I say that? I can actually, in the next slide, I think I can give you a picture of that. And I thought I would add this picture. Now, I remember, just to show, is a bit more of a dramatic picture here. The other picture was based on electron microscopy. This is based on atomic force microscopy. This is that same map, but for a photosynthetic bacterium, purple bacteria. And you see on the upper orange colored panel there, those rings, the big rings contain the reaction center. The little rings are the light harvesting centers, and they're these incredibly symmetric, beautiful structures. It was the first structure that was solved, the first light harvesting protein whose structure was solved at high resolution by Richard Cogdell in Glasgow. That was his life work. So, just give you a physical sense for why this is intriguing, is we can solve this problem, simple simulation with, say, 1,000 molecules in a 2D lattice with one trap. And we know that, we know a couple of parameters here in the lifetime, how long we have to move that excitation. It's not infinite. It's not like electrons moving in a transmission wire for a power grid. We have a few nanoseconds to do this. And we know the size of the antenna complex. We know how long it takes for each of those excitations to jump from chlorophyll to chlorophyll, which is a few hundred femtoseconds. In any case, as I said, it will take about 2,000 jumps until we reach that trap on average. And the total distance traveled is amazing, 2 microns. But the diffusion length is only 30 nanometers in this simulation, because it's a random walk. It goes forwards, but it also goes backwards. It goes in the right direction, goes in the wrong direction. So here's the problem. The question is, so imagine now, coming back to the synchronization, if we could direct that excitation, this would make a huge difference. And studying different photosynthetic systems is an amazing range of organisms in nature. I'm sure we haven't even explored in detail the tip of the iceberg, but this gives a really nice example. In fact, I think Bob Blankenship brought this to my attention. This is a slice of mud, a few centimeters thick, taken from Yellowstone National Park, from the hot springs. And they're full of photosynthetic organisms. And you can see in the photograph of the slice on the left, different colored stripes. And on the right, I've made a little cartoon of those going from the top, what's exposed to the sun, down. Diatoms, cyanobacteria at the top there. There's your purple bacteria coming down here. And on the left there, I'm showing the spectrum of the sunlight, at the surface. Then after it goes through the diatoms and cyanobacteria layer, after it, here in the third figure there, after it goes through the purple bacteria layer, which absorbing the near infrared, you're not left with a lot of light. A little bit in the near infrared, those green sulfur bacteria are photosynthesizing effectively under moonlight conditions. Yet they live. And this is what fascinated people after the experiment that I'll show you in a couple of minutes, from Graham Fleming's group. I do point out, though, that these guys aren't prolific. You're not going to see them grow. Okay, it's not like kelp, which can grow 10 centimeters in a day. These might, in some cases, these will only reproduce every few months or a year. But nevertheless, there are these questions, especially after Richard's structure came out, shown on the right there, this beautiful arrangement of closely packed light-absorbing molecules, chlorophyll-type molecules, called bacterial chlorophyll. And there was a conference in Lithuania, 1996, right at that time, perfect timing, very exciting meeting. Very cold too, I remember, for the time of year. Nobody had packed appropriately. But from the abstract book here, the major concern at that time is the time over which excitation can be considered coherent. And why would one ask that question? Is it coherent or is it not coherent? Well, as I said, entropy beats us. You can organize, imagine keeping in sync excitation on these 18 chromophores in that red ring of the complex. Doing it by hand, it's a little bit like putting your fingers in a dike. It's physically thinking about it, trying to choreograph all these oscillators or pendula, keeping them in sync. They do get out of sync. They may have slightly different frequencies and so on, as energetic disorder. From that time, in any case, what researchers really wanted to do is, you were asking, well, how can I do an experiment in any case that would give deeper insight into this synchronization, quantum synchronization or coherence? And this is a famous picture from the Engel-Fleming paper on that, on the complex that was isolated from that bacterium I showed you photosynthesizing under moonlight. And the experiment they did, I can say qualitatively, is a little bit like this. So you see in the absorption spectrum I've shown on the top right with the little purple square. Purple square is highlighting two peaks that you can see. So when you cool the protein down, you see that even though these chromophores, the molecules are identical, they're in slightly different protein environments. So they have a little shift associated with that. So we can pick out, and I'm going to simplify this slightly. You can pick out two of these chromophores, you see them there, and the laser pulse, which is that gray thing, is very broad compared to any of these absorptions. It can't frequency resolve any of this. It just hits the whole thing like a hammer indiscriminately. And so what that's a little bit like would be if we had two different frequency tuning forks and bang them both on the table together. So not one at a time here, which would be narrowband excitation. We bang them both together and listen. When you do that, as you know, you hear not just the two musical notes, but you hear a low frequency beating, the frequency difference between the musical notes. I'm sure we've all done this in primary school. And this is the idea of this experiment, is to bash the complex optically and listen for that beating. And that would tell you that those two molecules are not uncorrelated, or they're correlated. In other words, they're synchronized somehow. Otherwise, you can't get those frequency waves to line up and give the beating. And so that's what this 2D map is. And so the white arrow from Greg's paper is pointing to what's called a cross peak here in the 2D spectrum, and made a movie of that as a function of time. And that's kind of the signature of that beating. And I'll show you in a simpler example that. So that's what got people excited. And to summarize is the key, and you see this, the fireflies are because you can get clusters of fireflies that synchronize themselves for other reasons, a nonlinear oscillator like the heart cells. They all flash in sync, but it's a good example of why this experiment works. What the challenge was, if we had a forest of fireflies that were not synchronized, and we could do an experiment that took an average over a cross section through that forest, like with a big beam of something. We couldn't see the fireflies individually, and we would watch them. and do a measurement, we would say the fireflies absolutely glow. But we would also say, well, they glow, but we don't know that they flash. They would just be a steady glow that we would measure because we're averaging. Some of them will be off, some of them will be on when we perform the measurement. We'll just take an average over that. So we can't resolve the flashing. Now, how would we resolve the flashing if we can't do an experiment that looks at each firefly individually? Well, the answer is if we can get them all flashing synchronously, then we can still take an average and we'll see the flashing. So get them all synchronized to start with. The amplitude of that flashing is all together, and we can measure that even if we average over 10 to 15 fireflies. And that's what these experiments are doing. You know, that's the big deal here. Doing it on an ensemble. So and summarize this a little bit, this is taking, we normally think of spectroscopy as measuring as a function of frequency, and we're mapping out the quantum mechanical resonances, if you like, of the gap between the ground state and the excited state. And molecules are complex, they have vibrations there as well. And that's something that has been done in incredible detail in the past and maps out that spectrum. Instead of doing that, there's a shift here with these short in time pulses that necessarily, because of the Fourier principle, carry a lot of frequencies in them. They look almost white for a 5 femtosecond pulse. We, instead of probing the energy gaps between these individual quantum levels, you see here on the famous particle in a box from my undergraduate lectures on the left here, we just excite all those levels together. Bang, that's hitting the tuning forks all together. And what that does, interestingly, is it takes us from this quantum mechanical picture of eigenstates that we're taught as an undergraduates to a picture that looks much more classical and is intuitive. It's a wave packet, it's still quantum mechanical, but it looks a bit like a particle oscillating in a potential backwards and forwards, just like we'd imagine a classical oscillator, a pendulum, or anything would do. So if this is a vibration in that molecule, which is what it is here, we excite the molecule to the excited state, and we excite a bandwidth, a number of these excited state vibrational levels, and then we see that in real time, that vibration, and we can map it out. Because you see the wave packet is resonant with the blue side of the pulse on the left there. You see that gap between the ground state potential and the excited state is big. When the wave packet moves to the right side of the potential, you see that gap is small. It's on the red side of the pulse. And so we could map this out with a white laser pulse. And on the right-hand side now, you see exactly what that looks like. So it's a broadband pump probe experiment. Those frequencies, wave numbers, are on the x-axis, and then time goes down on the y-axis. And so those ripples are the oscillations. It's seeing the wave packet going backwards and forwards. The node, that white line, for instance, that you see at 16,000 wave numbers on the left, that's when the wave packet passes through the middle of the potential, which does twice per period. And so it doesn't show up on the plot here. That's not at that frequency. And instead we get a change in sign of the ripples as we go from the left to the right side of that node. And so that's really watching in space and time that wave packet. So some amazing technology here, and pretty simple to do, that particular experiment. Okay, so just coming back to the beats between the tuning forks, what is that? Is it profound? Is it just because there's different frequencies being radiated? And not that interesting? Well, the key is that the frequencies radiated here have to be synchronized, and there's some reason for that. This is from a really old paper, and there's, I admit there's really too much stuff here in this equation. I should rewrite it maybe and simplify it. But the diagram, but I can explain it pretty easily. The diagram on the left here shows you the principle. One ground state, we just have to worry about the G on the left here, and two excited states. They're the two different frequency bands, say we saw in the FMO complex, and I'll show you another example in a moment. With our if we had narrowband laser pulse, we could pick out state A, or B, and we would know exactly that we'd excited that level. With the broadband pulse, we don't know which level we excited. So in quantum mechanics, this is where it gets interesting. It means we've made a superposition. That's that top equation, the Schrodinger's cat, alive plus dead, because we don't know which level we excited. Now, when we probe what's going on here, we could probe that level A and say, oh yeah, well, 50%, it looks like there's 50% chance we excited that, and the same with the B. And that's the same as the classical expectation. If these levels are connected, or if these are not, we have no idea. But if they're synced, which means we have this coherent connection, in this case, because they're in the same molecule, we additionally get this interference in the detected intensity because of this superposition, and that's this cosine function here. So remember why that is. It's because in quantum mechanical probability, we don't sum probabilities or sum rates, if you want probability per unit time. We sum amplitudes. And so we sum, and the amplitude is that superposition state there. So if we go back to that top equation, if you like, we get the probability of A, that's C squared A, plus the probability of B, that's C that's C squared of B, just squaring the coefficient, just think of it as a number. And we get the cross term, 2 times CA times CB, and that's the interference. And that's what gives those oscillations, because it carries this exponential thing that turns into a cosine, is all it is. Okay, so let's have a look at a clean example here of, just turns out this is nanocrystals in solution. It doesn't, the spectroscopy of this doesn't matter. Why the experiment works well here is because the sample, the electron micrograph on the bottom looks a bit messy, but their widths are all almost identical to atomic thickness, and some of them are sideways. That little blue arrow points to them. It gives us nice sharp transitions. So the two electronic states, HX and LX, that's like your A and your B from the last example. And this is the 2D spectrum, just like in the Fleming paper. And we look at the cross peak, which is where the olive and the blue lines intersect, okay, either at the top or the bottom there, which tells us, for instance, we get that, for instance, if we photo excite the HX state and probe the LX, it's just like a map, okay, reading out coordinates, the input light, the output light. But the signals that get there are a little bit more, there's a bit more interesting stuff in there than just that. But in any case, you see the beats in the right-hand plot there and those oscillations. And so there's a point here, and it lasts very long, 13 femtoseconds. And this is electronic coherence, for sure. And this is what we expect. We know we expect this because if you have a look at the width of the absorption lines in the spectrum on the left-hand side, I mean, it's hard to be calibrated here on the fly on a talk, but they're reasonably broad as usual. And there's this direct relationship here between this dephasing time and that width, because the width tells you a bit about the distribution of allowable energy disorder. Okay, and this is what beats us. And I think I might have a picture of that coming up from a classical point of view. Quickly, I'll just show you an analogous experiment from the blue proteins pulled out of these algae, called cryptophyte algae, another photosynthetic organism, which are invisible but everywhere in seawater and lake water. And again, we can map out the different colored molecules in this protein. Now they're chemically different. It's a bit easier to do it, I might say. And as I said, I'm not getting into I don't need to walk you through excruciating detail here, except to say, if you look at the black absorption band on the top right, you notice that qualitatively there seems to be two big bumps there, and there are actually. And on the low energy bump, the red absorbing part corresponds to absorption from these 4 red molecules you see in the structure. And on the high energy side, the green the other bump is due really to the absorption from those green molecules. And so we can do the same kind of experiment, excite this with broadband excitation and look at that. Did we synchronize the red molecules with the green ones? Okay, which would be interesting observation. They're 20 angstrom apart. Okay, this isn't, you know, a strongly interacting molecules. We would not expect them to be synchronized. And so this is the surprising result, which I'll explain very quickly and clearly. But you look at the cross peak on the bottom right of the movie, it's definitely doing something. It's the amplitude's jumping up and down over these few hundred femtoseconds of the movie. And that frequency that it's jumping up and down at is, of course, the frequency difference between the two bands you see spread along the diagonal, which was the band from the red absorbing molecules, its absorption band, and the one from the green absorbing ones. So it all fits here. It's the beating mentioned between the two tuning forks, but now the musical note, you know, it's not just a semitone apart, it's much bigger. So what were we seeing though? It turns out, as a long story short, stimulated by Fleming's work and so on, what we realize now is that vibrations built into those molecules, not coincidentally, let me just go back to this picture, not coincidentally, are exactly resonant with that gap, electronic gap between the red and the green molecules. Same goes for the chlorophylls, but this is a bit more obvious here. And by analyzing these data that I just showed you in some detail, what we could show is that that oscillation, which lasted a really long time, much more than 13 femtoseconds, was because the molecules physically are vibrating in the wave packets I showed you earlier. And that quantum mechanically mixes with this electronic synchronization, keeping the excitation on both molecules together in some sense. It's a bit different than the initial ideas. And that's the picture that's emerged. But I think the summary here is, if we think about coherence here, what does it mean? It means the excitation would be both on the red and the green molecule together. And that does lead to interesting quantum correlations. What it means is that if we think of the absorption classically or semi-classically as oscillations of the electrons in the molecule, that oscillation is synchronized on the two, okay, even though the frequencies are quite different. And that is allowed by the coupling between the molecules. So not like in the Plaraton case where the molecules all coupled to light. Here the molecules are coupling to each other. It's like two pendula connected by a light spring, and that keeps the two pendula moving synchronously together, or the famous example of Huygens clocks. What beats this is disorder. So if the two clocks, if one keeps time well and the other is really hopeless, then a light spring won't keep those pendula synchronized. They're going to get out of sync. And this is the problem with energy disorder. I mentioned the energy disorder. That's why Fleming could do the experiment in the first place, remember. It made the absorption bands, those molecules, quite different. The averaging here, and just think of this as a cartoon. It's actually a density matrix for an excitonic system that might look like this. But it makes a point, even if you've never heard of a density matrix, is that if we take one snapshot of what's going on here, put the information into the Hamiltonian of the system and solve Schrodinger's equation with everything fixed, even when there's a fair bit of disorder, what we would see here in this particular diagram, any dark blue squares that are not on the diagonal are a signature that quantum mechanically we have this coherence. And you see that on the top left. Now what we do is we have to take an average, and each of these successive plots actually is ensemble averaging more. So the first one is only 5 copies of the system, and then 10 or something like that. Yeah, 5, 10, 20. But look at this, even after averaging only 5 copies of the system, not 10 to the 15 yet, just 5, we've lost any of that quantum information. That's just from this disorder effect. And this is what defeats a lot of this synchronization, obviously not in the polariton case. So there's ways to get around this, which I think is the interesting forefront here. But this is the issue as soon as you average. So let me, the last few minutes, I'll tell you an interesting classical example. It's the complete opposite. And this might give us a hint, some direction to go in. So this is, I mentioned Huygens clocks. So I think he designed very accurate pendulum clocks for the Navy. I suppose the Dutch Navy, maybe I should check that story again, but it's important in maritime in those days. And he was ill in bed one day, in any case, looking at his 2 clocks that were on a shelf. Or you can think of it here in the photograph. It's a mock-up of them hanging together. And what he noticed is that they weren't identical, the 2 clocks. They were pretty close. And in any case, no matter how he started those pendula, he got out of bed, he wasn't that sick, I suppose, and put the pendula at different starting positions. No matter how he did that, they always ended up synchronizing with each other. The reason for that is that the oscillations of the pendula are coupled weakly through the beam that those clocks are hanging from. So that's the light spring I mentioned before that couples them. And so on the left, see a simulation here of the model that explains this and explains the synchronization of the heart cells, the fireflies, and anything else we can think of. Thousands of oscillators. I've started half a dozen of these oscillators out of sync, very weak coupling between them. They all have different frequencies, by the way. It's like the experiments I just showed you, where we lost synchronization in 13 femtoseconds. But look what happens here. We get in sync. It's amazing. And the ensemble average is shown on the bottom. So, and the ensemble quantum average I just showed you, we lost. When we started out synchronized, we lost that very quickly. Here we gain synchronization. And these are nonlinear oscillators. It's engineering. It's just a feedback loop here. And that it turns out that it's the parameters of the system are such that the energy is lowered by keeping the pendulum moving together because of the forces between them. And this is an example of engineering synchronization. Very small interactions, though, can do this. And it's the way that the pendula interact that matters. I'll show you a simulation of it for fun here. What the picture here is, the axes, I've said speed relative to the mean. That means in a rotating frame. In technical terms, in the position there, it's a phase plot. It's telling us of the phases of the oscillator. So it's going to tell us if these dots are all bunched up together, that represents everybody going up and down together. Okay, the more you spread out, like you see the dots here, that means everybody's doing their own thing, regardless. And so that's how we're starting the simulation. You just run it. This is just watching the synchronization happen in time. Gradually, those phases lock together, and now everybody's oscillating together. Very simple model, a mathematical equation of motion on the right there. So the simplicity is the key. You can synchronize clocks even when there's a frequency spread in them, even when most of them don't tell the right time. We can get them to synchronize if we couple them. So in those experimental examples, I did stress that everyone tends to get out of sync because of their different frequencies. It's a good way to think about it. It doesn't have to be the ensemble frequencies are different. It can be that the frequency fluctuates. It's just as powerful. And this is called decoherence and dephasing, respectively. And this is, of course, the challenge for this field at the quantum mechanical level, and it makes complete sense. entropy increases. Remember, this is like 100 years ago now at the start of the talk, I told you about that light matter state, the polariton of the molecules in the cavity, and I pointed out that that low polariton state had very low entropy. Well, that's not something that can be sustained. It can be engineered, but it's not where the universe will end up. On the other hand, we can beat entropy with energy. This is this is why the atmosphere stays around the Earth. For instance, it's held there by gravity. That entropy would have it dispersed throughout the universe, all those gases. And so this is the driven oscillators down the bottom. I think it's an interesting discussion point. So, and that's classical synchronization. The quantum synchronization has been much less explored in these ways. I think it's fascinating. If we could do this, it would certainly allow us to think about new ways to control or regulate systems at the molecular scale, and not just be a slave to these random fluctuations of noise that I pointed out very early on in the talk. So the traditional view might be, well, we need to work out how to beat this dephasing and decoherence. How do we get around entropy? And one way might be, to take inspiration from these driven oscillators. On the other hand, we might take a point of view, if we can't defeat entropy, maybe we can exploit it. And that's something I've looked at very recently with those polariton systems, which is a photonic kind of structure. There are photonic structures, by the way, in some photosynthetic organisms, very interesting ones. We don't really know exactly what they do. Just to throw that out there, maybe for discussion. But if we can't defeat it, maybe we can exploit it. So I didn't have a picture of that, but that's something I just published in JPhys Chem Letters, if anybody is interested in that. And so I think that's a good place to stop, so we have plenty of time for discussion. Of course, I thank the funding agencies and my students and postdocs who've contributed to all of this, and thanks again for hosting me.